So it's such an incredible honor to be the recipient of the Elizabeth Hay Award. I really don't have words to express how humbled and honored I am to be receiving this award today. And in part, it's because it's from a society that I truly love, and I feel like I've grown up here. So I remember myself, maybe about 10 years ago, being a graduate student and going to SDB for the first time to give a major talk. And I've always just grown and fell in love with this community of incredibly welcoming and creative minds. And I think it's been so influential for all of my life. And that's particularly why it means so much to me. But as a recipient of this award, I really had the occasion to get a chance to know Elizabeth Hay a little bit better. And I hope to convince you that it's really kind of serendipity that I've been awarded this award, and we have a lot more in common than you may imagine. And so, as John had mentioned, Elizabeth was a pioneer in cell and developmental biology. She actually loved the electron microscope. This is Elizabeth at 80 years of age in front of an EM. And I really felt inspired to get a chance to know a little bit more about her. And so I did a little bit of digging. And so this is Elizabeth's own words. I went to a meeting in 1954 in Atlantic City with some friends just to go to Atlantic City. So I think this reflects the fact that besides being just a tour de force cell and developmental biologist, she probably was rather cool as well at the time. But what I didn't expect was her next sentence. And my gosh, Keith Porter gave a symposium lecture on the fine structure of the substance of the cytoplasm. It was composed of Palladi granules that Palladi and Porter had just described. And I came back, and wow, from then on, it was electron microscopy for me. And I found that to be incredibly special. This was Elizabeth as a very young medical student, and I think it influenced the trajectory of her career and her science. And it's also influenced my trajectory as well. And I actually dug up a picture of probably the type of image that Elizabeth saw when she was at this meeting. And this was actually in 1954. It was submitted in 1954 by Palladi, for which he won the Nobel Prize. And he described these very, very tiny dots that coat the entire crevice of a cell's cytoplasm as a small particulate component of the cytoplasm that turned out to be the ribosome, which is one of the most spectacular molecular machines that I think have ever been created to translate genomes into proteomes, and for which he won the Nobel Prize. Elizabeth then went on to think about the directions that she wanted to take as a medical student, and surprisingly, she applied to Palladi's lab to basically study these particular structures and was rejected. So it's sad that she didn't wind up studying these directly, but she became very close friends with Palladi throughout his entire life. And I just wanted to fast forward sort of decades further, and just to illustrate how much cryo-EM has really evolved, and EM in general. And so it's now possible to take a cross-section through a cell, to take a very small sliver, and to freeze it, and be able to get to near atomic resolution of these Palladi bodies. And not only did it fascinate Elizabeth initially, but this has been a point of obsession for both me and my lab. So typically, if you ask an undergrad student to basically draw a typical cell, they usually completely neglect to draw these tiny, beautiful molecular motors and structures. And part of the reason is because, as you can appreciate here, they really seem to be these tiny particles that all look the same, and sort of situating every single square crevice of the cell's cytoplasm, and simply blindly sitting there, taking in any of the instructions of the genome in the form of an mRNA template, and translating all the final effector proteins required for every single key cellular decision. But I hope to convince you that in your favorite cell, which these machines are located, they're not simply blindless rogue-like entities, but rather play incredibly fascinating roles in instructing key cell fate decisions. And just to orient you, there's upwards of 10 million ribosomes per cell, and that ribosomes account for almost 85% of the total RNA pool and approximately 1/3 of the cell's dry mass. So a couple of years ago, research in my lab, and as John mentioned, largely from, and I'll talk about the history of this a little bit, developmental biology had enabled us to really come to a different view of what these machines might be doing. And instead of thinking of the textbook view, that again, they might be all the same and sort of produced in a similar way, research in our lab started to suggest that perhaps there's more than meets the eye. And instead of thinking that all ribosomes are the same, potentially there could be hundreds of variants of ribosomes. And illustrated here in different colors are the components of the ribosome. So they're comprised of 4 RNA species and approximately 80 core ribosomal proteins. And work in our lab has suggested that perhaps ribosomes might be different either by virtue of changes in the composition of these core ribosomal proteins, their post-translational modifications, as well as their functional activities. And that, in essence, we might want to return to our favorite cells and now zoom in into the millions of ribosomes that exist there, because they may not be just fly nanomachinery that are taking in the genetic code, but instead these changes might influence the spectrum of mRNAs that are translating to proteins in our favorite cell types and really execute a more tailor-made proteome of cell differentiation, development, organismal development, as well as the execution of cellular decisions. And so I just wanted to very briefly tell you how I became fascinated by ribosomes. I don't think as a developmental biologist I would have ever thought that I would be studying ribosomes. What I can tell you is that as a graduate student I was extremely passionate about learning about how body plans are patterned. And as all of you know, this is sort of a most extreme version of this. This is sort of a very profound pattern that can apply, where the misexpression of antennapedia, a homeodomain transcription factor, converts an antenna into a leg. And I instead became obsessed by this little mouse. And it doesn't seem to have as grotesque of a malformation, but I became excited because it was a mouse that spontaneously arose in the 40s in a highly inbred strain of animals. And over the decades, it kept getting passed along, largely because it had this funny looking pink tail. But I became really excited when we examined the body plan of these animals. So while they seem seemingly normal, when you zoom in on the exoskeleton, it's full of very profound patterning defects. The most dramatic are approximately 20 homeotic transformations that line the anterior and posterior axis of this mouse mutant. And this is just a zooming in of the cervical region. So for example, here you know that your cervical vertebrae normally don't have pairs of ribs associated with them, or at least I hope they don't. And here you can see in this mutant, there's a full pair of ribs that are associated with the 7th cervical vertebrae. So that 7th cervical vertebrae really thinks that it's a first thoracic vertebrae. The 6th cervical vertebrae in the wild-type animal has this characteristic anterior tuberculum, and now that same shape or morphology to this vertical element is present on the 5th cervical vertebrae in this mutant. So in collaboration with Toshiko Shiroshi's lab in Japan, we positionally cloned the gene responsible for these profound defects in body plan patterning. And I think it was such a huge surprise to realize that it was a single component, a single ribosomal protein that belongs to the mammalian ribosome, RPL38. And I think this was the first link between a core component of this machinery and something as fundamental as establishment of the mammalian body plan. And this really obsessed us because we were thinking it would be a transcription factor, a chromatin modifier. This was really nothing like we had imagined. And so how was this ribosomal protein doing this? So the first thing that also came as a very big surprise, and I actually had undergrad students when I started as a fellow in my lab, who tried to understand whether this core subunit of the ribosome showed any developmentally regulated pattern of expression. And for a long time, I had to convince students to actually do these experiments because they just assumed that according to the textbooks, this ribosomal protein should be expressed in all cell types. It's a boring housekeeping protein. And so I still remember when my undergrad ran in and she started to make section and feature hybridizations and also looking across all vertebrates to look at the pattern of its expression. And what was quite remarkable was that RPL38 was not ubiquitous, but rather showed expression patterns that were enriched in specific tissues. And so I'm going to direct your attention here in the mouse, which is a sagittal section through the developing mouse embryo, where we can see that RPL38 is a beautiful marker of developing mouse somites. And what was exciting at the time is that not only was RPL38 expression restricted, and now we have done sort of more sophisticated mass spectrometry analysis that I'll tell you about in a moment, but this expression pattern really mirrored where loss of function phenotypes were evident in this animal. So it suggested to us that somehow an RPL38 containing ribosome might be important in translating a specific code that would be required for formation of the body plan. And so how is it doing that? We were obsessed to understand the answer. So basically we went all the way back in evolution. We went to yeast and we realized that without this ribosomal protein that was thought to be a core component of the ribosome actually functioned quite well. So it could translate the majority of the genome without any problem. However, using translational profiling, we were able to instead uncover selective subsets of transcripts that really required this ribosomal protein for their translation. And this included subsets of homeodomain transcription factors that, as you all know, are critical for anterior and posterior patterning along the apiaxis of the developing axial skeleton. And so this sort of brought us full circle because it led to the identification of targets that might be responsible for the really profound homeotic transformations that were evident in these animals. So the next task was trying to understand how this was molecularly or biochemically achieved. And I was very lucky to have my first graduate student, Shifeng Shui, who actually won the Harold Weintraub Graduate Student Award for her research on this subject. It was a really big black box. I don't think it's still fully understood, but the question is, how can a ribosome with a specific constellation of proteins recognize specific target mRNAs? We knew that a ribosome containing RPL38 was being built up in developing somites, but how is it translating only selected classes of mRNAs? I think in terms of looking at our genomes, one observation is that the RNA layer of gene regulation is probably one that has the most dynamic ability to fold and bend, making secondary and tertiary structures. And what Shifeng was able to identify was that a common theme to all of the target mRNAs being translated by RPL38 containing ribosomes was the presence of this intricate structured RNA element that we termed an internal ribosome intracyte-like element because it resembled the mode of regulation in viral mRNAs that also required structured RNA elements for their translation. And Shifeng was able to show that these elements that seemingly weren't there just by looking at the sequences of the 5' UTRs were really critical then in instructing body plan formation. So I'll show you an example of how important this is in a moment. So we actually went in and we were able to take out this very small RNA regulatory element within the 5' UTR of specific subsets of Hotch genes. This one is within HOXA9. And you can see that there's no change in the overall transcript levels. There's no change in the boundaries of Hotch gene expression. But if we look at the protein level, despite the fact that a transcript is born, it's normally localized. Without this UTR element, it really cannot get translated faithfully in time and space. We basically cannot detect the HOXA9 protein without the presence of this RNA landing pad for a specific type of ribosome. So this was exciting and suggested that during evolution, a specific class of ribosomes may have evolved to translate specific subsets of genes that were so critical for the mammalian body plan. When I started at Stanford 3.5 years ago, we had a dream. Our dream was to try to understand how many different types of ribosomes may actually exist in every single living cell. So if a cell has 10 million ribosomes, how many ribosomes might be different? Can we develop genome-wide tools to be able to ask this question? And secondly, can we understand, using new technologies, what these ribosomes might actually be translating? So this was a really risky question because we didn't know if RPL38 was just an oddity or there was more in terms of the composition of these ribosomes that might change, for example, during the course of cell differentiation. So I really have to thank two of the most talented postdocs who joined my lab to take on this effort. I really, as a junior investigator, could have never imagined to have such two talented junior scientists in my lab who were bold enough to address this question. And I'll be speaking a little bit about their work today, but it has both two very important studies have come out just recently in the past couple of months, and I strongly hope that this will inspire you to read them. So the first thing is work that has emerged from Jen Shi in the lab. So it may seem like a relatively simple question, but because there's 10 million ribosomes in a cell, how can we get an absolute measure of the absolute amount of each core ribosomal protein in a ribosome? That was a huge technological challenge that Jen undertook. And what she was able to do is use a very sophisticated mass spectrometer called the triple quadrupole mass spec, and to use selective reaction monitoring to, for the first time, get at this question. And this was achieved because she can purify translationally active ribosomes from any of your favorite cells. But in addition to loading these samples onto this mass spec, she can have a metric, and in this case, use heavy peptide labeled standards for each of these ribosomal proteins. So we have an absolute metric or quantity to measure the endogenous components too. This enabled her to the first time be able to get a more quantitative picture of the absolute abundance of specific classes of ribosomes within cells. And in collaboration with Irv Weissman's group at Stanford, we're very excited to basically develop a roadmap of ribosome heterogeneity across very important and distinct cellular lineages. So I'll first start with an embryonic stem cell. So this is a ground state pluripotent cell. And I'll show you her initial findings looking at the absolute quantities of ribosomes that are translating actively the ESL genome. So the first thing that Jen found was that there are certain core ribosomal proteins that belong to what she's termed as class 1 proteins. So these are ribosomal proteins that exist in all ribosomes within embryonic stem cells. But then she's slowly building a huge catalog of class 2 ribosomal proteins. So these class 2 ribosomal proteins we know are sub-stoichiometric. We know that they're heterogeneous. So for example, RPLP1, which is a major component of the large subunit of the mammalian ribosome, is only present in approximately 37% of all ribosomes that are actively engaged in translating the ESL genome. So while these technologies are exciting, they've been incredibly difficult to build up, Jen wanted to take this one step further. Now that she can pinpoint heterogeneity within cells, she wanted to develop a systematic way of being able to understand what these ribosomes were translating genome-wide. And to be able to do that, we needed the advent of CRISPR-Cas9. Because with CRISPR-Cas9, she was able now to endogenously tag different heterogeneous ribosomes at their endogenous locus. So she hasn't perturbed any of these ribosomes, she just endogenously tagged them. And then using a technique known as ribosome profiling that was developed initially by Jonathan Weissman's lab at UCSF, she was able to read out what these ribosomes might be translating. She can pull them out, IP them out from the rest of the pool of the ribosomes in the cell, and see at a codon by codon resolution what, and if anything different, do specific classes of ribosomes translate. And this is a typical experiment that Jen has done. This is for the RPL10A containing ribosomes. And Jen sort of, when she first got these results, was so excited. She came to my office, her hands were shaking, and I was asking her, is there any preference in what these ribosomes are translating? And Jen showed me these results, and we were quite taken aback. So basically, here we have an RPL10A containing ribosome. We'll profile the transcripts that it translates, compared to the total ribosome pool in the embryonic stem cell. And using very stringent fold change cutoff, Jen really wanted to pick the targets that really stood out as being the most differentially bound by this ribosome. She found hundreds of transcripts that either were depleted from these ribosomes or were enriched from these ribosomes compared to the total background ribosome pool. When she stood back, we started to immediately get excited and started to ask additional questions. The first question is, if you compare two pools of ribosomes where we can get these footprints, are they randomly translating different transcripts? Are they the same? Are they different? What might be special about them? And so here are categories of transcripts. They're not random because all of these are statistically significant geoterms of networks of mRNAs that are unbiasedly shown to be associated with specific functional categories. So for example, if you compare these two different ribosome subtypes, an RPS25 heterogeneous ribosome or an RPL10A containing ribosome, you can see that the enriched targets for this heterogeneous ribosome tend to be translating mRNAs critical for cell signaling, while this ribosome tends to be translating mRNAs that are critical for cell cycle. And why that's very exciting, and I'll show you this here, is we really try to now go in transcript by transcript and try to reconstruct the logical circuitry of how these genes might be functioning together. And so here are all of a huge subset of mRNAs that are being translated by this RPL10A ribosome that I just showed you. So not only do they functionally segregate in translating important components of the cell, including cell signaling, they really are remarkable in the sense that we've been able to identify entire signaling pathways where every single signaling component from the reception at the cell membrane to the reception at the nucleus, every single component is being translated by one type of ribosome. So similar to sort of the operon system in bacteria, I think we are witnessing perhaps a much greater intricate balance of what's being translated by these different ribosome subtypes. And I think it's also giving us very exciting hints into what this could mean for a cell, what this could mean for an organism. And so I just wanted to highlight some interesting groups. So if we look at these RPL10A ribosomes, let's zoom in on one network. One of these networks are enriched for genes that are critical downstream responders to growth factors and mitogens. So these include insulin growth factors, the early growth response genes, and if we look at the mRNAs that instead are depleted away, so mRNAs that are not being translated by this ribosome, we actually see an enrichment of gene networks in the complete opposite category. So these are some of the cell's biggest stress response and cell death pathways. And so we think that perhaps there's intricacy in how genes are converted into proteins, by which even a small change in one of these core subunits of the ribosomes can elicit a very rapid remodeling of how networks of mRNAs will be translated upon certain moments in the life of the cell. So for example, if you wanted to tell a cell to really start to proliferate very fast, I would suggest you dial up its RPL10A containing specialized ribosome subpool so it can start to immediately translate these mRNAs. And one of the exciting things that Denise's work has further suggested is if we look at all of these different ribosomes within subcellular space, there might be further intricacies in terms of what they're translating in the three-dimensional architecture of a cell. And Denise's work has been able to now trap ribosomes at specific subcellular localizations. So for example, ribosomes at the ER that Palade had initially described. And Denise was able to further show that there are hundreds of additional RNA binding proteins that bind to such ribosomes, and that one single RNA binding protein can specialize these ribosomes for selectively translating mRNAs that are destined to be secreted. But I became even closer to Elizabeth because one of the most exciting categories of specialized ribosomes that we're following up on are actually with respect to what gets made outside of the cell. So this is actually a transmission electron microscope made by Elizabeth Hay herself, and this is a cross-section through one of her cells. And you can see here there's these beautiful depositions of collagen fibrils that she was able to identify and have been so inspirational, as John mentioned, to her career. She really discovered the ECM. And I just wanted to illustrate to you perhaps that this ECM is being built up by specific types of ribosomes. So one of the specialized types of ribosomes that my lab has recently identified is one that seems to be tuned to translating ECM components. So this is its network of mRNAs. And in the center here in green, you can see examples of these ECM components, including multiple components of the collagen superfamily, laminins, catepsins, as well as matrix metalloproteases that are faithfully translating or turning out very selective programs of proteins that are being secreted from the cell and are critical to the extracellular matrix. And so, of course, in 20 minutes, it's hard to tell you about all of our work, but I hope that the examples that I've shown have inspired you to think about the millions of ribosomes in your favorite cells. I don't think that they're there and just floating in the cytoplasmic soup. They're doing really important things, and I think it's taking a lot of new technologies and a lot of new methodologies to be able to probe this layer of gene expression. But if I step back, I really believe that we can parallel our understanding of what is emerging to be a potential ribosome code with that of a histone code. So decades ago, histones were viewed as very, very boring proteins. They were thought to package the DNA and have very little regulatory specificity in how our genomes were expressed. And then slowly it became noticeable that histone tails, as well as histone variants, and their post-translational modifications really play a remarkable role in epigenetic control of gene expression. What our research is suggesting is that perhaps another spectacular molecular machine, one of the most ancient, has parts that are interconvertible, and that each of these components play a very important role in allowing or endowing the translational machinery to translate specific targets in time and in space. I think this also is reflective of a new layer of control on the RNA-mRNA template itself. So I started off by telling you about these Hox mRNAs that have these intricately folded RNA structures. Although I don't have time to show you, all of the specialized ribosomes that I have described today, our initial work suggests that this is a mechanism by which they latch on to their target templates and really requires a completely new set of tools to be able to understand how the RNA structure is folded into shapes in vivo. And my expectation might be that decades from now, instead of searching for linear sequences that you sit in front of your computer and you blast for your favorite sequence in a DNA, that's really going to be RNA shape, these landing platforms for which ribosomes can more intimately interact with their target transcripts that will give this fundamental increase in specificity in translational control. So I'd like to end here, and I really wanted to acknowledge the mentors that have been so fundamental for my own growth. So I think John mentioned that Elizabeth didn't even apply to Harvard in 1948 because she was told that female bathroom facilities didn't exist yet. But in 1960, she became a professor at Harvard. In 1975, the chair of the Department of Anatomy. And I think she enabled great women scientists to come after her. And I think I have a long lineage of women scientists that have been inspirational to my own growth. I'd like to particularly thank my former graduate student mentor, Lee Niswander, who really just sparked my love of developmental biology. She's such a passionate and exceptionally wonderful mentor that I think she knows that I would have never been a scientist had I not met Lee. And also, likewise, it was great having Gail Martin when I started off as a UCSF faculty fellow. She was my neighbor and instrumental in my growth there. So with that, I'd like to end. I'd like to thank all of the current members in my lab. As John mentioned, we kind of started this in an unbiased genetic forward screen. We were passionate about understanding the mammalian body plan, and it kind of transformed our understanding of gene regulation, and it kind of reflects the current state of current members in the lab. We have such a multidisciplinary group. We have chemists, developmental biologists, RNA structure folks, as well as hardcore computational scientists. I'd also like to thank the wonderful environment at Stanford. I'm so privileged to be in the Stanford Developmental Biology department, which I think is one of the best departments in the world, and has just been an inspirational place to start off my lab in. And I'd also like to thank key collaborators as well as funding sources that have made this research possible.